In this video, I'll begin to discuss the molecules of life. Those are the biological molecules that we normally think of as being common to all life on Earth. First, I'd like to just begin by having you start thinking at a molecular level. How many molecules are there really in a cell? If you took a single cell, for instance, from a plant, and expanded it by about 1 million times, 10 to the 6 times, it would be about the size of an auditorium or a classroom, depending on the cell type. And within that room, then, you would have a nucleus, which would be about 10 feet to 12 feet in diameter, depending on the cell. And a nucleolus inside of that would be about 5 or 6 feet in diameter. And then a mitochondrion would be about the size of a person, although it would be a rather heavy person because they're a little wider than a normal person. Although mitochondria often are convoluted, so it would have different arms and different branches going off in different directions. A chloroplast would be about twice the height of a man. And probably 3 or 4 diameters of a person. If a bacterium were to invade this cell, it would be approximately the size of a heavy person also. And then viruses would be about the size of a tennis ball, or an average virus would be that size. The walls surrounding the cell, the plasma membrane would be about half a centimeter in thickness, coating all the walls and the ceiling and the floor, and completely continuous with proteins embedded throughout that membrane. The cell wall would be approximately as thick as a cinder block wall in a building, so that's about equal to the same size. The nucleus also has 2 membranes, and it's actually a single membrane folded back on itself. So it would be about 1.5 centimeters thick, and it would have nuclear pores dotted throughout it, which would allow certain proteins and other components into the nucleus, and it would be exporting RNAs and some other things out of the nucleus. The DNA within the nucleus would be about the thickness of a thick string, and the RNA polymerase on that would be about the size of a lima bean. And then the RNA that would come off of that transcription unit, our RNA polymerase, would be about the size or thickness of kite string, so thinner than a thicker twine. And then you have ribosomes, so the mRNA would wind up in the ribosome. Ribosomes are about the size of a golf ball in this scale. And in a typical bacterial cell, there would be 20 to 60,000 of those. And in a typical plant or animal cell, a eukaryotic cell, you'd have anywhere between 2 and 6 million, and sometimes on larger cells, even more than that. So imagine all of those in this cell. There are a lot of ribosomes. A large protein would be about the size of a lima bean, a smaller protein about the size of a pea. And in this scale, you would be able to see an atom as a sort of speck of dust. So you could see like sodium ions and chlorine ions floating around in the room. So now imagine this cell, this room as a cell, about 2 million golf balls, the ribosomes floating around, hundreds of thousands of proteins, hundreds of thousands of mRNAs and other RNAs floating around, as well as ions, a couple of chloroplasts, several mitochondria, perhaps some viruses and bacteria infecting the cell. And so they're just packed. In fact, the cell, instead of being just a bag of water, is nearly crystalline. So if you just take a little bit of water out of that cell, it would begin to crystallize and lose its function. So now if you take all those components and in this room and then begin shrinking that down and shrinking that down 1 million times, then you would not be able to see it with the naked eye. You could perhaps see a single cell in a dissecting scope and certainly in a compound scope. But if you take something around 25 cells, 25 by 25, then you could see those as a little fleck of, let's say, skin or something on the desktop, and that would be about the size of a building, right, if you blew that up a million times. So that's at the level we're working at when we look at these cells and all of the components inside of them. Okay, so let's start talking about some of these molecules. The basic structure of a nucleotide is this, where you have a pentose sugar and then attached to a phosphate group, and then a base, one of these nitrogenous bases, a purine or a pyrimidine. So here's a little more detail about that. Here's the base, one of the nitrogenous bases. The purines are adenine and guanine, and the pyrimidines are cytosine, thymine in DNA, and uracil in RNA. Here's a pentose sugar. It's marked as 1', 2', 3', 4', and 5' carbons, and those become important in some of the other notations later. And then a phosphate group linked together in an ester bond, a ester bond here between the 5' carbon and the phosphate. And one other difference between RNA and DNA is RNA has a hydroxyl in the 2' position, and DNA has simply a hydrogen in that same position. So here are the two pentoses, ribose with a hydroxyl at the 2' position, and 2-deoxyribose or DNA with a hydrogen in the 2' position. And it seems like a minor change, but this hydroxyl in ribose means that the same packing is not available, is not possible for ribose as with deoxyribose. And also that hydroxyl is a reactive group, so that makes RNA much more reactive than DNA, which is not as reactive at all. And here are the 5 nitrogenous bases, adenine here, guanine here. These are the 2 purines, and here are the markings for each of the carbons. There's 1, 2, 3, 4, 5, 6, 7, 8, and 9 here, and 1, 2, 3, 4, 5, 6, 7, 8, 9. And then in cytosine, just single ring structures for the pyrimidine, cytosine, thymine, and a difference here in having a methyl group for thymine and just a hydrogen there for uracil. And here are the markings for those rings, 1, 2, 3, 4, 5, 6, and 1, 2, 3, 4, 5, 6 there. Here's a ball and stick model of the same bases as well. Here's adenosine, and the star is where it's attached to the ribose or deoxyribose sugar. Here's guanine again. Position 9 is where it's attached to the sugar. And then there's cytosine, thymine, and this methyl group would be a uracil, would be hydrogen in uracil, and they're attached at the N1 positions to the sugar groups. Here are some of the bases with their phosphates attached in DNA, so a couple of base pairs in line. So you can see here's the 5' phosphate carbon going through the 3' sugar, again a phosphate through the 5', and the 3' hydroxyl at the end. So this is why we say that DNA is always listed in the 5' to 3' direction, and there's a reason for that. That's how it's polymerized also. And these two strands would be an anti-parallel, that is from 5' to 3' in this direction, and from 5' to 3' in the opposite direction. So they're anti-parallel, parallel but going in opposite directions. There's the sugar phosphate backbone, which is hydrophilic, and then the bases are on the inside and they're hydrophobic. And between a GC pair, you have 3 hydrogen bonds, and between the bases in an AT pair, there are 2 hydrogen bonds joining those together. So in DNA, the GC pair forms a stronger bond than does an AT pair. Just to show you how these sit with respect to one another. So the guanine, cytosine, the base pairs would be on the interior of the molecule. And then here's where the sugar groups, the deoxyribose here and the deoxyribose here, these are in what are called cis positions because they're on the same side of this molecule. If they were trans, then one sugar would be there and one sugar would be attached over here. But in DNA, they're in the cis form. If we flip to a T pair, then you can see that the molecules kind of sit on top, one on top of the other. And they pack quite closely together in the DNA molecule. So again, there's the next base pair up from that. And then on top of that would be this. So you can see how those base pairs and those bases sit one on top of the other in the middle of the DNA molecule. Again, here are the base pairs. There's an AT pair, and the length of the hydrogen bond in each is about 0.29 to 0.3 nanometers. And there are two such hydrogen bonds. And then in a cytosine guanine base pairing, there are 3 hydrogen bonds ranging from 0.29 to 0.30 nanometers. Now, there could be a hydrogen bond between this carbon and the oxygen, but the distance is too far between those. And one more time, we have this sugar phosphate backbone, 5' end, 3' end down here, hydrophobic region with the bases in the middle with the hydrogen bonding a GC and an AT pair, and then another phosphodiester bond going from the 5' end to the 3' hydroxyl end on that side, on the other opposite molecule. These molecules also are called self-complementary, so every time you have an adenine on one side, you will find a thymine on the other side, and every time you find a cytosine on one side, on the other strand, you'll find a guanine, so that you can have a mechanism for copying these by using the template as one strand to copy the opposite strand. DNA can take on many different forms, but the usual DNA is what's called bDNA, and it's a right-handed helix. That is, if you take your right hand and curl your fingers around, then your thumb would be pointing upwards, and that's the direction that the DNA would be coiling. So this one has a major groove here and a minor groove formed by those indentations moving into the bases. And proteins often attach only to the major groove and not to the minor groove. And these are just 3 different diagrams of what that looks like. Although DNA and RNA are both nucleic acids and have some superficial similarities, their forms and activities are very different. So RNA takes on a wide variety of shapes, and it can be an enzymatic molecule as well. It doesn't form double helices for any length of time, but forms double helices and then folds out to form other structures and holds certain nucleotides out to become reactive groups as well. So it's sort of, you can think of it as almost like acting like a protein. One feature of mRNAs and eukaryotes is that a cap is added to the 5' end of the mRNA, which attaches to a cap binding protein and also aids in halting any sort of degradation that occurs on that end of the mRNA. So it's usually a 7-methylguanosine that's added, and it's added not in a 5' to 3' direction, but in a 5' to 5' triphosphate bond to the end. So it's a very special feature to mRNAs. Another feature of RNAs is that the bases can hydrogen bond on 3 different sides. So there's the standard Watson-Crick edge in each case. Here's A, G, C, and U. But also it can hydrogen bond to other bases by the Hoogsteen edge, as well as the sugar, what's called the sugar edge. And in doing so, then you can have many different conformations of the RNA, how it folds up between each of the different bases in that RNA chain. So there are actually 12 geometric families then, 6 ways of connecting: a Watson-Crick to a Watson-Crick edge, Watson-Crick to a Hoogsteen edge, Watson-Crick to a sugar edge, Hoogsteen, Hoogsteen, Hoogsteen sugar, and sugar, sugar, and each in the cis or trans conformations according to which side the sugar group is attached. The rest of the chain is attached. So there are quite a few different possibilities of how the pairs can go together. And also there are non-standard pairs, so it's not just an AU and a GC pair, but you can have AC pairs, GU pairs, and other types of pairs also. And in addition to base pairs, there are base triples in RNA. So here's a UAU base triple, where the A and the U have formed a standard Watson-Crick edge, but then the A has also paired with another U along the sugar edge and the Watson-Crick edge of the U. And then here's a CGG pair, base triple, which is, these are actually quite common in RNA. There's CGG as well as CGC base triples that are common in RNAs. There are some very important fundamental molecules also that are made of RNA. One of them is NAD and NADH, these nicotinamide adenine dinucleotide. So this contains an adenine and a nicotinamide moiety in the molecule, and this is important in what are called redox reactions. So this one will transfer electrons along with often a proton from one molecule to another. This is one of the fundamental molecules that is active in oxidative phosphorylation and photosynthesis and many other processes in the cell. And this one down here is cyclic AMP, which is an intracellular messenger. When this is formed, it will message proteins and other molecules to become activated. And another one that isn't shown here is ATP, the one that transfers phosphate groups and high energy in a lot of different reactions in a cell. ATP is an RNA molecule. RNAs contain not only A, G, C, and U, but they contain many other bases. One here is N6-isopentenyl adenosine, 6-methyl-adenosine, quiosine, iosines here. And here are some other bases. Inosine. Inosine is interesting because it will pair with any other base, A, G, C, or U. Xanthosine, 7-methyl-guanosine, which I already showed you, pseudouridine, 4-thiouridine, dihydrouridine, and 5-methylcytidine. Some of these are important, especially in tRNAs, ribosomal RNAs, and other types of RNAs as well. In total, more than 120 different bases have been found in RNA, whereas only 6 are present in DNA. So the 6 in DNA are A, G, C, and T, but adenosines sometimes are methylated, so that's a 5th one, and cytosines are sometimes methylated, so that's a 6th one. But over 120 bases in RNAs, which shows you, which indicates anyway, that they can be much more versatile in their functions. And they are. Some are ribozymes. They fold in dynamic ways, meaning they change. Some transfer energy. Some participate in redox reactions. Some synthesize polypeptides, that is the ribosomes. Some deliver amino acids to the ribosomes, those are the tRNAs. Some contain coded messages that are decoded in the ribosome to form proteins, those are the mRNAs. And then many control gene expression, usually by shutting it down or controlling which mRNAs are allowed to be expressed. So just to show you how some of these come together to make proteins, the mRNAs, and then contain the messages. They're capped with that 7-methyl-guanosine and then fed into the ribosome. The ribosome consists of ribosomal RNA, which contains the enzymatic function to attach the amino acids together in the polypeptide and proteins. Those come together in the small and large subunit along with proteins, which primarily hold the RNA in its conformation. And then the tRNAs carry the amino acids into the ribosome, matching up with the anticodon on the tRNA with the codon on the RNA, the mRNA, to place the proper amino acid in the protein polypeptide sequence. So all of these functions then are being performed by RNAs, not by the DNAs. The DNAs are primarily the holder of the genetic information, but it's really the RNAs that do most of the work to produce proteins. So that brings us to proteins and the peptide bond. In biological systems, only L amino acids are used. In a few cases, D amino acids are used, but by and large, they're all L amino acids. It could have gone either way, but for some reason during biological evolution, only the L amino acids were started with and continued. Each amino acid has what's called an alpha carbon, to which the R group is attached, and the R group differs depending on which amino acid. And then there's an amino end and a carboxyl end, and that's why you have amino acids. They're connected by means of a peptide bond between the amino group on one amino acid and the carboxyl group on the other amino acid, and that joins then two different R groups together on those alpha carbons. In doing so, a water molecule is released. In the ribosome, depending on the mRNA and the tRNAs that are attached to the amino acids that are being brought into the ribosome, a genetic code is used. And so this is the genetic code according to the codon in the mRNA. So here's a GUU encodes for a valine, a GCA encodes alanine, etc. We know from some evidence that this bottom line, those were some of the first amino acids used in the genetic code back when life first got started, when translation first got started. And that some of the last amino acids that were added to the genetic code are up in the upper right-hand corner, the more complex amino acids. The proteins that are made via translation consist of many, many different categories, including structural proteins like microtubules, enzymatic ones such as lysozyme, signaling proteins such as insulin, transport, storage, regulators, kinetic proteins, that is, ones that move things like actin and myosin in muscles, receptors like rhodopsin that will receive light and make a certain chemical reactions based on the light and the wavelength of the light, special purpose proteins such as antifreeze proteins, recognition proteins such as immunoglobulins, and others. So these different categories of proteins can be found in many different locations in the cell. Enzymatic ones such as DNA polymerase, RNA polymerase, structural proteins such as microtubules, receptor proteins such as those in the photoreceptors of the chloroplasts, and many, many other signal proteins, etc. Here's a diagram just showing a couple of different proteins as well as some RNAs too. So the RNAs are the ribosomes and the tRNAs. But then here's an antibody Here's an actin filament in a muscle cell, and some other enzymatic proteins or small globular proteins. Here are two viruses, and then the proteins that make up those viruses. So those would be purely structural on the outside of those viruses. So they vary quite a bit in structure, size, and function. So as cells went from being primarily RNA-based with small peptides into DNA-based using proteins, they could become a lot more complex because the DNA would be transcribed into RNA, which would include a code that would produce the proteins that could have much more broad-ranging functions than could the RNA or the DNA. So the next molecules I want to talk about are cell membranes. They're made up primarily of phospholipids that form a bilayer, so there's a hydrophobic center to the membrane and then hydrophilic sides on the inside and the outsides of the cells. These form the cell membranes around bacteria and eukaryotes as well as organelles. The membranes in archaea are slightly different, and I'll discuss those in a different lecture. These molecules have hydrophobic tails, primarily formed of fatty acids, although they're usually isoprenes in archaea. And sometimes they have a double bond, which forms a kink in one of these two legs of these molecules. They're connected together with a glycerol molecule, a 3-carbon molecule. Two of those carbons are attached to the fatty acids, and the other one is attached to the polar head. The head group is always polar, so it's pointed inside of the cell and outside of the cell in an aqueous environment. They're attached through a phosphate into this other part of the head group, which is polar also. In this case, it's an amino group. In some cases, it's other molecules that are polar as well. These membranes can be disrupted by detergents and soaps, which is one of the reasons that you use detergents on your clothes and dishes and soap on your hands. This will kill most organisms because it essentially solubilizes their membranes, and so they can't survive without that membrane. Here's a diagram just illustrating that fact that when you use soaps and detergents, you disrupt the membrane. The DNA and all the contents and proteins and everything else then leak out. And then they are also surrounded by other detergent and soap molecules, which then solubilize all those things so that they can be washed away with water. Incidentally, early in evolution, fatty acids formed spontaneously. So phospholipids probably could be formed quite easily very early on Earth, so that the possibility of building a membrane was there from some very early moments in the evolution of life on Earth. The last set of molecules I want to talk about are carbohydrates, sugars, and polysaccharides. These also form, some of them form spontaneously, and so these were probably available very early on Earth as well. So here's some of the monosaccharides, fructose and glucose, and then disaccharides, including lactose, which is, everyone knows as milk sugar, but also builds up in your muscle cells when you work hard. And then sucrose, which is the normal sugar that you have, table sugar. Then you have these polysaccharides such as amylose, which is in plant cells, which are just repeating units of glucose. You would find amylose in plant cells like potatoes and things like that, storage sugars. The main storage sugar in animals is glycogen, so this you would find in your liver. And so you would store glucose units in your liver this way, and then when those glucose units are needed, they are chopped off off one by one and put into glycolysis. Another polysaccharide is cellulose, which forms the main cell walls in plants, and these are just repeating units of sugars as well. So next I want to talk about how some of these molecules essentially form part of the major pathway of gene expression, that is, going from DNA, transcribing the DNA into RNA, and then translating the RNA into protein. And this is known as the central dogma, proposed by Francis Crick in about 1956 or 7. But I want to go beyond the central dogma also. So here it is in a nutshell, the flow of genetic information in modern cells. You have DNA, which gets replicated. DNA is transcribed by RNA polymerase into RNA, and then the RNA, the mRNA, is fed into a ribosome for translation into protein. However, in an evolutionary sense, this scheme cannot have evolved in the same order from DNA to RNA to protein. And I've already told you that it looks like the first organisms consisted primarily of RNA and some peptides. So DNA and proteins weren't a part of that system at that time. So if we put it together in an evolutionary sense, then RNAs here, those were the first biological processes, were probably based on RNAs and probably some small peptides as well. Next, the RNAs became more elaborated, and then the first ribosomes evolved. So now you had true protein synthesis. So the proteins, the peptides actually became longer, and then they were called proteins because they're over 50 amino acids long. So now we have a system consisting of RNA plus proteins that includes ribosomal translation. And this probably occurred right around 3.8 to 4.1 billion years ago. And then finally, by about 3.5 to 3.8 billion years ago, a variant of RNA, that is DNA, began to be used as the genetic material. And it was much more stable, so the genomes could become much larger because DNA is much more stable than RNA. RNA is prone to many more mutations than is DNA. And because of that, an RNA-based organism would have much smaller genomes, and its progeny would die much more often due to the high rates of mutation. So let's look at how this whole process occurs. Again, we have DNA being replicated by DNA polymerase, and then DNA being transcribed into RNA by RNA polymerase, and those are actually related molecules, related enzymes. And then RNA is being translated into proteins by ribosomes, which are primarily RNA-based. So let's start the process by looking at the DNA. Its single function is to store the genetic material, and it needs to be replicated at high fidelity with few mistakes. So that's part of this process. Next comes transcription, but transcription actually results in several different things. In eukaryotes, you have transcription by RNA polymerase 1 called POL1, and that only transcribes ribosomal RNAs, and specifically the large molecules of ribosomal RNA. Those RNAs are then folded, attached to some proteins that hold them into their proper form, and that becomes the ribosome. Next, there is RNA polymerase 3, or POL3, which transcribes tRNAs as well as some smaller RNAs, which I'll mention later. Amino acids are added to these tRNAs by certain protein enzymes that add those amino acids to the tRNAs. And they do so specifically, so a tRNA that has a certain anticodon will have only a particular amino acid added to it, so it's very specific. This is where the genetic code really comes from. Next, RNA polymerase II will transcribe only mRNAs, and so that one's specific for messenger RNAs. Those are processed into mature mRNAs by adding that cap, the 7-methylguanosine. If they have introns, those will be cut off, and often a poly-A tail will be added to them as well. These all come together during translation, so the ribosome feeds the mRNA through it, and the amino acids are brought in by the tRNAs to produce polypeptides, which are the proteins. So the part that is the central dogma that everyone learns is only a part of this, right? So DNA is transcribed into mRNA, and then that's translated into proteins by these ribosomes and tRNAs with the amino acids attached to them. But all too often, students are taught that everything becomes mRNA, and that's actually not true at all. In fact, it's only a small component of all of the RNA in a cell. The majority of the RNA in a cell actually makes up the translation machinery, the ribosomal RNAs and the tRNAs. And then these proteins, which are produced by translation, go on to do all of the cellular processes, many of which feed back into these other processes. That is, they become DNA polymerase, they become DNA binding proteins, they become RNA polymerases. They're involved in processing and glycolysis and many other tasks also and functions in the cells. And this isn't everything that happens either. So you can see in here there are other arrows, which I won't talk about too much, but there are other arrows here that become SNORNAs, which are small nucleolar organizer RNAs that aid in the processing of ribosomal RNA. There are microRNAs over here that control expression of the mRNAs that have already been transcribed. And then there are these silencing RNAs that attach to other RNAs over here and keep them from being expressed also. There are also a couple of other RNA polymerases in plants that do perform other functions to control the expression of certain RNAs as well. So you can see that RNAs are extremely important in the cell and are responsible for making the proteins that are also important in the cells and creating the diversity in the cells and in the organisms themselves. And this really only scratches the surface into what RNAs do in a cell. So this is only one set of processes that involve RNAs, and there are many, many others. So in general, RNAs can be messages, that is mRNA, and this is the one that most people focus upon. Structural, including parts of the ribosomal RNAs, tRNAs, and others. Enzymatic, including the large subunit of the ribosomal RNA or the ribosome, introns and others, and controlling, small RNAs and others. They can also exchange electrons, they can exchange energy, etc. So there are other functions for different RNAs as well. This table shows some of the proportions of these polymeric RNAs within a cell. So here's ribosomal RNAs. By mass, it makes up about 80% of the RNA in a cell. About 10 million molecules are present in a typical eukaryotic cell. Only 4 to 6 different genes, though, so it's fairly homogeneous. tRNAs plus other small RNAs make up about 15% of the RNA in a cell, about 20 million molecules, and 20 to 30, sometimes up to 50 or so different tRNAs and other small molecules. The mRNAs make up only about 5% of the total RNA in a cell, about 350,000 molecules in any particular cell. But there are 20 to 30,000 different mRNAs encoding different proteins. And it can be as low as 4,000 and as many as 50,000, depending on the species. Therefore, about 95% of these RNAs are involved in the protein producing process. But most of the complexity in the cell, the RNA complexity, is within the mRNA molecules themselves. So that's the end of this video on some of the important molecules of life, going from the very earliest organisms that were primarily based on RNA and peptides up till the present, where organisms use DNA as the genetic material. Most organisms do. And RNA for a variety of processes, and these processes began probably between 3.5 and 4.2 billion years ago, but still exist in all of your cells as well. Somewhere around 3.8 billion years ago, then the first organisms that had DNA in them used DNA as the primary hereditary material. Then the genomes could get larger, including our very large genomes. And that was transcribed into RNA, which was still used for the same variety of processes as in the original cells. And a lot of that RNA then became mRNA that was then translated into proteins. And then the organisms that could diversify due to the variety of proteins it could produce really became very successful and spread out as the entire tree of life that we see today. Okay, that's the end of this video. I will talk to you later.